could not be more delighted that the Broad Institute is a host for VIZBI. This is one of the most important things going on in biology. And I mean that, in a surprisingly serious way. Right next door, when the Whitehead Institute was built, David Baltimore, brilliant Nobel laureate, one of the great scientists of his generation, in designing the Whitehead Institute, did not initially see any need for accommodating computers. At the last minute, they altered the construction on the second floor to put in a computer room. Because in those days, the data you had was the data in your lab note. It was what you printed out from the spectrophotometer and scotch taped into the lab note. It was observations you wrote down in pen. The thought that most of the data that you would use in interpreting your experiment would be somebody else's data was so completely unimaginable. And yet today, I can't think of an experiment that we do here at the Broad, or frankly that's going on across the street at MIT or down at Harvard or anywhere else in the world, where the interpretation of the experiment doesn't use massive amounts of data generated elsewhere. This is a sea change. The Human Genome Project was a part of that. I think it was the first time biology said, we're going to go get ourselves organized to collect lots and lots of data and make it freely available. And I got to tell you, this question of how to represent the information of the human genome was itself already a complicated question. When the draft sequence of the Human Genome paper came out, there was a lot of attention to this fold-out in Nature of the human genome sequence, called a roll fold in magazine speak. And so a lot of attention went to representing the genome on a roll fold that people could put up on their wall. It was a good thing, but it was a pretty easy thing. Because in those days, the genome was a linear thing. There was a line, and we didn't know that much about it. You could write the nucleotides if you had a very little pen. You could write the density of different kinds of repeat elements. You could put positions of genes, things like that. It wasn't visually, it was, there were some interesting challenges, but nothing like what has happened since then. What has happened since then has been driven by the collection of vastly more data. As you all know, but I'll say anyway, the explosion of new technologies for creating sequencing data, just as one example, all sorts of new machines that for us have given us the experience that from 1999 to 2006, we were so proud that we went from generating 1 billion bases of data to 70 billion bases of data. And then as new technologies came in, the next 2 years looked like that. The next year looked like that. The year after that looks like that. We generated 125,000 billion bases of data last year. It is very likely that this year we will get maybe another order of magnitude, or at least a factor of 8. It blows my mind to imagine the amount of data we're getting. Now, if it was data that fit really nicely in some paradigm, that'd be one thing. We wouldn't need VIZBI. But it's not. You see, these data they don't fit on our line so well. They're letting us generate all sorts of maps. You get any question you want. If you can figure out how to convert it to, say, a sequencing question, you can generate huge amounts of data. So you can take the data, the supposed genome, and start layering stuff on top of it. But you very quickly realize you're in trouble.
There is no one human genome. We differ, not just by single nucleotides, which you could figure out how to represent, put little lollipops and say this could differ between, an A or a G, but some of us have insertions here, some of us have deletions there. What are we going to represent that on top of? Oh, and it's not just that we have differences. We care about the fact that those differences are correlated with each other. So the local correlation of those differences, I don't know if there's a pointer thing. Yeah, there is. Oh, that was a bad idea. Okay. Is that yeah, there is. So local correlations matter a great deal. So this was the original roll fold of the genome with different things represented. But we have differences of nucleotides, insertions, deletions, correlations amongst those things. But then we get the different species, the chimp, the mouse. Well, they rearrange the genome in different ways. Chunks are missing, chunks are rearranged, chunks are duplicated. How do we navigate such an evolutionary picture? How do we look at the picture, not just of how the pieces match up, but how well they're conserved across evolution? How do we layer on top of this knowledge of transcripts when they vary in many different ways? There can be 20 different transcripts, some of which might use these bases, and on other bases, some of which could be coming this way and reuse those bases on the other strands. Oh, and then there's chromatin maps. There's more than 5,000 different chromatin maps that have been constructed today, different chromatin marks in different cell types laid across the genome. And then there's associations with disease. There are hundreds of diseases for which people have looked at millions of genetic markers and gotten different degrees of significance. How do we see those things? And how do we see simultaneously, when we're doing a disease study, that over here we have some markers? Yes, there are markers, there are actually two different alleles that cause the disease, because these guys are correlated and these guys are correlated, but they're not correlated with each other, but they're in the same region. Two alternative disease-causing haplotypes. How do you represent that, of which guys go together? How do you glance at that and at a glance see that there are evolutionarily conserved patches over here? And oh, those patches, they have certain chromatin structures in liver, and your disease might have something to do with liver, which might mean going out to the literature to find that out. It's no longer even a question of the fact that it doesn't fit on the line because the line has insertions and deletions. It's fitting across a space of many tissues, many organisms, an entire literature. And then it gets worse than that, because you start putting functional data on it. You take a genome-wide RNAi library and start knocking out genes. And you say, yes, if this gene over here were knocked out, it affects 48 other genes across the genome who together form some ensemble of things whose pattern is seen again in these kinds of settings. I mean, obviously, if I knew to ask that question, I could write a compute to do that. But I don't even know to ask that question. I want to see that question presented to me. And then there's circuitry. As you begin to start knocking things out, you begin to start building circuits. Well, look, a lot of the alleged, circuits that people write are big hairballs right now. They're not really very useful. How are we going to represent, now we've totally lost the line, circuitry? Compare circuitry, see the rewiring of circuitry. This is really tough and important stuff. This is not what in 1985 I think folks thought about, which was when you're done with your science, you'll find an illustrator to help you draw the figures. It just isn't that. It's before you start your science, your colleague who thinks about visualization is part of the scientific team. Indeed, your colleague who thinks about visualization is a respected innovator in science in at least 3 different dimensions. There's enormous creativity that's going to be needed. There's creativity with regard to functionality. I think about it the same way I think about algorithm designers. The person who came up with the binary tree sort, the divide and conquer sort, made an unbelievable contribution to the world beyond the person who had the, inserted into the list sort. N squared versus n log n guaranteed was transforming the functionality that came from that algorithmic invention. There is a question of functionality where the visualizer is in effect going to be an algorithm designer, making it quick and easy to connect things up. But there's metaphor also. Metaphor is so powerful in science. When we talk about a promoter or an enhancer for a gene or something, we often forget that we don't quite know what we mean by an enhancer. But we have this word, the enhancer, and we have some idea in our head. Indeed, we have a picture in our head, not just an idea in our head. It's always about a picture. And those pictures are incredibly powerful because we come back to those metaphors. The visual scientist is going to invent metaphors over the next 5 years that are going to be powerful organizing tools for science going forward. And they will affect thousands of studies because the metaphor, the visual metaphor, will have been created. And then, frankly, there's just beauty. That things that are beautiful have a huge communicative power as compared to things that aren't. So visualization will combine this functionality, this metaphor, and this beauty in a wonderfully creative domain that is going to be part of our science from the get-go. It is going to be transforming. It will, the same way I think the Genome Project is powerful because it produced things that can get used in 10,000 ways. Visualization tools produce things that can be used in 10,000 ways, and you won't be able to control or even know or even imagine what those impacts will be. It is a powerful integrator, and it's an integrator not just for scientific research, I would say. Certainly, it is an incredibly powerful component of scientific research, but it must be a powerful tool for education as well. We are reaching the point now where there is no excuse for teaching the way we used to teach. It's just inexcusable to be doing 21st century science with 19th century teaching. I teach intro bio. I use the chalkboard. It's a powerful tool because it slows me down to the rate at which students can learn, et cetera. I like the chalkboard. But I know that with the right visualization tools, one could do amazing things in teaching. Students could teach themselves in amazing ways at their own pace. They could see things I can never draw on my chalkboard. I don't use PowerPoint in my 701 intro bio class because I don't think PowerPoint is my answer. I find it soporific. You're not involved in it. But my resistance to PowerPoint in a lecture is not a resistance to visualization being essential. But that visualization will have to be paired up with a map of learning. It'll have that students, what they have to learn and how they need to see the things to do it. It'll have to be paired up with not passive visualization, but active visualization, where you fly the controls of various different sorts of things. And so these tools together, converging both on research and on education, I think can have a huge impact. And nowhere is this more needed than in biology. Why? Well, biology is the largest research investment in the United States that we make in terms of basic research. It consumes $30-odd billion at the NIH, plus more at the NSF, plus more at other organizations. It's a major investment. Also because biology is the bleeding edge right now. It is the messiest data. You look at the heavens, and I'm sure an astronomer can tell you why the problem is hard, but for the most part, it does map onto the heavens. Right? There isn't polymorphic heavens up there where you have to worry about this patch is there one day, not the next day, or something like that. Biology has the messiest problems. It also has problems where we're constantly interacting and tweaking so that we have to be able to represent dynamic things. The astronomers, for the most part, are working on timescales where they can't really run experiments and their dynamics are kind of slow. So it is where the most interesting problems are going to arise. Anyway, I'm very excited about the fact that the Broad Institute is committed to being one of the homes for VIZBI. I think we're going to alternate or something. Certainly, as far as I'm concerned, VIZBI has a home here any and every time it wants to. It is central to what we are doing. I want to just close with a very brief comment on international exchange rates, if I can for a second. A picture is worth 1,000 words. This is a very serious problem. That may have been fine for some Europe, but if a picture is only going to be worth 1,000 words, we're screwed.